You found the skeleton. How would you tell it's a duck? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
Hey there, YouTube. As promised, we're looking again at Answers in Genesis craziness. I do want to note that because of technical difficulties with my editing software not doing speed adjustments the way I want, the sound sync is a bit weird here. I hope that's not too big a problem.
All right, so next one. Ken really likes platypuses, so that's why I picked this article. Mapping the platypus genome, how Earth's oddest mammal got to be so bizarre. But it doesn't really tell you that.
It's not bizarre. It's a beautifully designed little creature. Why would they call it bizarre?
Because it has no close relatives and has a highly restricted range, so most people will never encounter anything much like it.
Yeah.
You know.
Well, I mean, it does have characteristics of a lot of different animals.
Nope. It has general mammal characteristics, monotreme characteristics, and platypus characteristics. It doesn't have characteristics from other animals.
Well, let me show you what I put. This is a slide I use for my kids' program I do called Dinosaurs and More with Buddy Davis.
So here, Ken is about to go through some alleged chimerical features of a platypus in rapid fire, but we're going to examine each claim in turn rather than letting Ham simply blow through them.
So a platypus has a bill like a duck.
In fact, no. The platypus bill is nothing like a duck's, except in overall shape. In a duck, the majority of the upper bill is a centered premaxilla, with the maxilla only starting towards the posterior end of the upper jaw. And of course, it still has the basic tetrapod lower jaw with a dentary, angular, cerangular, and articular bone on each side, and an articular quadrate joint. Also, the covering of the bill is keratin, not skin. In a platypus, the premaxilla and maxilla run almost the whole length of the upper jaw, and the premaxilla is not joined in the center, but splits to either side, forming a U shape that curls medially at the anterior end. The lower jaw consists only of the dentary, and only has a dentary squamosal jaw joint, like all other true mammals. It also has a bill composed of soft flesh rather than keratin. You see, if the bill of the platypus really were like the bill of a duck, that would actually be a problem for evolution, because we should never expect to see a bird skull on a mammal. That would basically violate the law of monophyly, and while we might not have to completely abandon everything about evolution, we certainly couldn't use much of it to explain the platypus. Unfortunately for Ham, the platypus bill is almost nothing like a duck's bill.
And a beaver-like tail.
It's not really that much like a beaver's tail, though. Both animals have dorsoventrally flattened tails that are fairly broad, but it's much more pronounced in the beaver, and in the beaver, but not the platypus, the tail is covered in scales rather than hair. Really, though, neither tail is particularly unusual, and kind of flat and wide isn't exactly some amazing feature that anyone would suggest is a vital apomorphy that we can use to reliably trace biological lineages. Heck, even the North American and European beavers differ in this, with European beavers having less pronounced flattening and a more narrow tail. So even within what Ham would consider a kind, there can be variation in tail shape.
And hair like a bear.
Platypus fur is no more like a bear's hair than it is like a rat's, a tenrex, or a wombat's. So I'm not sure why Ham picked bear here. Basically, it's just dense fur with underfur and guard hair, like virtually all mammals have. But there is in fact one unusual aspect of platypus hair. It's biofluorescent. So if you shine a black light on a platypus, it'll glow like it's at a rave. Bears don't really do that. So even in the most basic trait, that the platypus have mammal fuzz, Ham isn't quite right.
Web feet like an otter.
Well, first, most webbing on all animals' feet comes about basically the same way. You see, all tetrapods have this webbing as embryos, but to separate the digits, the cells between them are usually programmed to die. All that has to happen for webbing is for that signal for the cells to undergo apoptosis, which is the technical term for programmed cell death, to simply be turned off. So ducks, frogs, Labrador retrievers, otters, etc. all have basically the same webbing for the same reason, because it's a simple mutation. Even some humans have webbing to various extents for this reason. However, you'll notice that in fact, no, the platypus does not have webbing like an otter. You see, like virtually all animals with webbed feet, the webbing of an otter does not extend past the unguals, that is the last bone on the fingers over which the claws grow. But in the platypus, the webbing extends past the claws by growing beyond and under them. That is unique in all animals. Once again, we have Ken Ham looking at only the most superficial aspects of the platypus. All it takes is just a few moments of thought to realize that his comparisons are nonsense.
Claws like a reptile.
Claws like a reptile? Like how? They're just standard amnio claws. The claws of reptiles and mammals are pretty much the same thing. They're both keratin sheaths grown over the bone core of the distalmost phalange, called the ungule. You could just say it has claws and leave it at that.
And it has hair like a bear.
I already covered the hair. I think Ken may be getting a bit forgetful in his old age.
And feeds its young on milk.
Yeah, it feeds its young on milk, because it's a mammal, which we already confirmed while checking whether the bill was like that of a duck. Also confirming it to be a mammal is the division of the vertebrae in the torso into thoracic and lumbar, which doesn't happen in other tetrapods than mammals.
And spurs like a rooster.
In fact, this might be the only fair comparison. The spurs of a platypus and a rooster are surprisingly similar. They are both keratin-covered bones that emerge near the walking part of the foot. But there are differences even here that are important. The bone spur of a rooster emerges from the fused metatarsal bones that are a characteristic of birds. On the other hand, for the platypus, the spur emerges from the tarsal bones proximal to the metatarsals. Also, for reasons we're about to get into, it is hollow, unlike a rooster's. So again, while this might be the most fair comparison so far, there are still significant differences that make these superficially similar structures quite different under the hood, so to speak.
And venom like a snake.
You see, once again we get to some oddities. While the action of platypus and snake venom are actually quite similar, the proteins that make them up are about as different as they could possibly be. And the platypus venom lacks the necrotizing effects of snake venom. So here we have two different kinds of venom that are doing mostly the same thing, but the proteins making them up are almost completely different. If a common designer uses common design for the same purpose, why don't platypuses just have snake venom? Perhaps minus the proteins that cause tissue necrosis. Why did the designer make it look exactly like you'd expect if snakes and platypuses both evolve their venom separately?
If it evolved from everything.
No. In fact, there is a fossil record for the platypus going from more generalized mammals to the specialized semi-aquatic weirdo of today.
Well, that's kind of what they're saying in the article.
No, they're not saying that at all. Look, the original article is linked in the description, and while one scientist did give a quote that I know in isolation could make you think that, you have to remember that scientists do not give their comments or write their research with young Earth creationists in mind, mostly because young Earth creation is so irrelevant to actual science that most scientists don't even know it exists, especially scientists outside the Anglosphere.
See, I think every time an evolutionist looks at the platypus, I think God smiles because I think he made it just for them.
I agree with you, actually. I think it was just to totally confuse people.
Except, it's not really confusing anymore. The platypus is a unique animal, but it fits neatly within the evolutionary phylogeny of mammals, with monotremes being sister to Theria, which itself includes the placental mammals and the marsupials. Nothing about it is chimerical, and its relatively basal genetics are in line with its fairly basal morphology, which is not actually that unusual outside of the skull compared to early mammals and mammaliformes.
But they try to say, well, this is where these different aspects that a platypus has emerged from. They'll say, well, it emerged from this. But there's no explanation as to how this information supposedly emerged, right?
Of course they explain how these things emerged, just not in that particular pop-sci article. Mutation, selection, drift, etc. are how this emerges. The fact that each and every summary of a scientific paper doesn't come out and state the very basics of evolutionary biology doesn't mean that it's not well understood.
It's just storytelling,
says the woman who literally bases her ideas about the history of the universe on a book literally full of stories. And no, it's not just storytelling. The dating is objective, testable, repeatable, and all creationist objections to the dating methods require the laws of physics to change or are simply illogical. We know mutations can and do add new chemical and morphological features to organisms, and that natural selection, sexual selection, and genetic drift occur. For the same reason that we know we can do a paternity test on a man and a child to see if they are related, we know we can tell if any two species are related, and we can even predict what the results will be accurately based on morphology of the modern animals as well as their fossil relatives. We can find expected transitions in a fossil record where we expect them to be based on the standard scientific methods of evolutionary biology and geology. None of this would be true if evolution were just a story. Sure, it tells a story, but one that, as far as we can tell, is largely true. And now let me tell you a story about the coolest people on the planet, my patrons and channel members. I'd like to just take a moment to thank my channel members and patrons, especially those pledging $20 or above: Ben Tovand, Ian Chen, Chris Love, Sphincter of Doom, and The Venerable Bede. My channel members and patrons help make this a much more stable income, because as you might know, ad income varies wildly from month to month, even with the same number of views. And it really helps this channel stay on the air. But if a monthly pledge, or a yearly pledge, which I have now activated, isn't right for you, there's a merch store linked in the description, and every like, share, and subscription really helps the channel grow. Also, those pledging $10 or above will have access to free 3D art assets, including materials and models, which will be made for Blender by me and can be used in any project without crediting me.
Well, this came 130 million years ago, and this came from 170 million years ago. And, somehow it got all these traits, and it's just storytelling.
And the great thing about this story is that we can correlate genetic divergence time tightly with the fossil evidence in many lineages. There is no reason that would be the case if evolution weren't true, since those dating methods and the actual fossils themselves are all independent and wouldn't track without evolution. If evolution were not true, we wouldn't see the fossils of ancient horses and rhinos diverging from being nearly identical to each other in every way through to their modern, quite distinct forms, all in line with the molecular clocks that tell us when we should expect them to have shared a common genetic ancestor, which then is also confirmed when the ages of the rock in question are tested radiometrically.
In other words, we've got a platypus, and because they believe evolution is true, they're coming up with all these ideas of how on earth this thing could have evolved, because they don't know how it could have evolved, because it couldn't evolve.
The evolutionary history of the platypus isn't really all that mysterious. Sure, there are better understood lineages, such as the hominids, cetaceans, or neoceratopsians, but there is indeed a fossil record of platypus-like ancestors, such as Montramatu pseudomericanum, 3 species in the genus Obdurodon, the not-quite platypus-like animals of family Steropodontidae, Steropodon and Taenolophus, which were more basal and didn't have the broad mouth of true platypuses, and we also have 5 other species of fossil monotremes that help fill in the picture of both platypus and echidna evolution.
Hey, you want to know an interesting thing about the platypus?
Mm-hmm.
Okay, Jennifer, seeing as you're an education expert, when was it first discovered?
Oh, the first platypus. I don't know when the first platypus was discovered.
1797.
Oh, okay.
In the Hawkesbury River, near Sydney, Australia. And you know what happened? They sent it back to England, and do you know what the scientists there did? They thought that somebody had cut up all these different bits and pieces of different animals and stitched them together. In fact, in the original specimen, you can see the scissor marks where they're looking for, trying to figure out where it was all stitched together and all the rest of it, because they couldn't believe such an animal could exist.
Yeah, because it was weird, and it didn't take long to figure out it was real and where it belonged in the Tree of Life, right next to the Therians.
Right. And it is amazing. Like, one of the things they say is how it lost these genes. Like, it lost the vitellogenin genes, which are important for the egg formation. Even though it does lay eggs, they're leathery eggs. They're not like a chicken egg. And so they say, well, it must have lost these genes. It didn't lose the genes. It never had the genes to begin with.
They weren't lost as in disappeared. They were lost as in they don't work anymore. They are what's known as a pseudogene. A pseudogene is a broken gene, but one that is homologous to an actual gene in some other organism. For example, humans have a pseudogene which they share in common with all other anthropoids for making vitamin C. All mammals have a vitamin C gene except for the few that have the homologous pseudogene, and all mammals have the pseudogenes for vitellogen. But in monotremes, they manage to hold on to functionality in one of these genes. So yes, we know for a fact that monotremes lost those genes, and they most certainly had them at one point.
Well, they're trying to say humans lost the same genes.
Again, because humans, like all mammals, have the pseudogenes. It's like if my car has a broken speedometer, but you insist that it doesn't have a speedometer at all, and it never had one in the first place. That's just a crazy person argument.
You obviously lost the genes for wings because you don't have wings.
Yeah.
So you've obviously lost those.
But they'll say they see remnants of it. A lot of times they'll say, oh, there's remnants of it there. But
Actually, she rather does have the genes for wings. As in, a human has essentially the same developmental genes as a bat. They just signal in different patterns and durations in embryo. This developmental genetic framework is shared by things as distant as fruit flies and humans. These genes are called homeobox genes, and the same homeobox genes are present in bugs and people. So essentially, nearly all animals more complex than a flatworm have the genes that in birds and bats result in wings.
Really isn't. There isn't any in human because humans never laid eggs. And the same thing with these. They never laid hard eggs. They've always laid these soft, leathery eggs. So
thing is, though, that humans do have pseudogenes for vitellogenin. Also, Purdom here seems to not know what vitellogenin is. She seems to think it has something to do with leathery versus hard-shelled eggs. The problem is that vitellogenin has nothing to do with the egg shell, and all to do with yolk proteins. You see, they are basically proteins that carry fat and sugar into the egg yolk. And, except for monotremes, all egg-laying animals have all 3 kinds of vitellogenin. And all but birds have soft-shelled eggs.
You know what I say to kids? It's designed to do what it does do, what it does do, it does do well, doesn't it? Yes, it does. I think it does. Do you? I do. I hope you do too, do you? So
okay, I'm glad you said that and not me. All right, very good. Okay, discovery boosts theory that life on Earth arose from a mix. So this goes back to the whole...
That glitch was theirs, not mine. But I want to say that I don't like that headline. There is no theory to boost. There is a hypothesis.
So this goes back to the whole origin of life, because this is a huge problem for evolutionists, right? They might be able to explain what happened after that, even though, again, the evidence doesn't support it. But they really don't know how it all came to be in the first place. So they're always trying to come up with ways to explain that.
Basically, what Georgia here just said boils down to, abiogenesis is an open question in science, so just say it was a miracle. That's not how science works. Open questions are investigated, whether creationists like it or not.
You know what? They're always coming up with ways like that, well, RNA and DNA and so on. But you know what makes life? You've got to have information.
Bingo.
And you've got to have a code system that reads the information.
Except that information can and does arise spontaneously. Every mutation does that. And we know that random proteins can in fact have function, although most don't. So random DNA sequences can code for functional proteins, and RNA can synthesize proteins without a ribosome. It's just not as efficient.
So just having bits of DNA or bits of RNA, that's not life.
No.
It's not life at all.
No one is claiming that bits of DNA and RNA are life in themselves, but they are important parts of life, being the molecules of inheritance. They are a necessary step in the formation of life as we know it, and so investigating how they can arise addresses the question of one of the vital steps in the formation of life.
You have to have already functioning units that can communicate with each other. And when we look at a human cell and you have all these organelles, and it's like they all function together, and they've tried bursting the cell to see if those organelles would function on their own, and they found out, oh no, they all immediately die when they're not together in this functioning unit.
Right.
So it's impossible.
Yes, now after billions of years of evolution and interdependence between organelles, they're really bad at functioning on their own, and really good at functioning as a unit. But for what reason would we think that this is necessary to all forms of life? The first life would almost certainly be far more simple than even the most simple modern cell, and it was also almost certainly less efficient at gaining metabolites and in its various biochemical processes.
And for a long time, they've said, well, maybe everything started with RNA. So you have DNA in the nucleus of your cells, and you make copies of it into RNA, which then gives the instructions for proteins. And so RNA is sort of the intermediate. So there's this whole idea of what's called the RNA world. That's what we started with, because it's a little easier to make in the sense that it's single-stranded and all this stuff. Well, the problem is, though, then you run into chicken and egg issues, right? Because to do all of this, so to get RNA or even DNA, you need proteins. But to get proteins, you need DNA and RNA.
You're wrong.
You're so wrong.
Wrong, 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 wrong. Wrong, 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 wrong. You're wrong. You're wrong. You're wrong.
No, you do not need protein to make DNA or RNA. That's the whole point of this article. Like, if she actually read it, she would know that. The entire discovery being reported on is that diamidophosphate, or DAP, can string together the building blocks of DNA and RNA, and even make hybrid DNA-RNA molecules. There is no chicken and the egg problem.
So they always have these issues with, well, which came first? And they're talking about this compound called DAP, okay, diamidophosphate, that it can string together bases of either DNA or RNA. So maybe we started with DNA and RNA. That's what they're going to say.
So here she just contradicted her previous statement. So which is it, Purdom? Is there a chicken and the egg problem, or can DAP string together DNA and or RNA?
But young people, a lot of them in schools would miss this, but you just read this one sentence. They demonstrated that a simple compound called DAP, which you just talked about, which was plausibly present on Earth before life, in other words, they don't know,
They have no idea.
before life arose, could have, which means they don't know, chemically knitted together tiny blocks of building blocks and so on.
There's lots of fuzzy words in here.
Yeah, it's all speculation.
Yeah, that's how science works. Now that we have a possible way to form DNA and RNA, we need further investigation to see if we can tell if DAP was present in the early Earth. But even when we reach the highest confidence in science, the researchers will still use tentative language, because science never proves anything.
Because
yeah.
they don't believe in God. They reject that. So they're saying, we've got to come up with somehow to try to figure out how life arose.
What you just said is one of the most insanely idiotic things I have ever heard. At no point in your rambling, incoherent response were you even close to anything that could be considered a rational thought. Everyone in this room is now dumber for having listened to it. I award you no points, and may God have mercy on your soul.
Nothing about abiogenesis requires atheism or agnosticism or in any way contradicts the existence of a supreme being. Abiogenesis researchers are not all non-believers, nor do all believers reject the science pointing towards the possible ways abiogenesis could have come about. This is just Ken Ham's persecution fetish showing through.
Well, and just because you can string together a bunch of bases doesn't give you information, right? You have to have a way to string it together in a meaningful way in order to get, and information only comes from an information giver, right? You have to have that in order to have this, so.
Too bad that's complete BS. Look, check the description for a paper about using random DNA sequences to generate proteins. And guess what? Some of them had function. So no, you don't need a mind to create functional proteins. Just randomly tossing nucleotides around will do that. Granted, less than 1% of such proteins are functional, but when you have a non-zero proportion of such proteins, and enough random DNA, and selection pressure, the percentage of functional versus non-functional proteins is going to skyrocket once reproduction and selection are involved. Anyway, that's all for this time. Please don't forget to like, comment, and subscribe. I'm the Dapper Dinosaur.